Okay, then I'm audible, no? Hello.
Yes, sir.
Okay, fine.
Yes, sir.
Fine. Maybe now you are second semester, and so whatever the syllabus initially I started for you, no, in when you are in a first semester. Okay, vitamins. That vitamins is moved from the first semester to second semester. More particularly, fourth unit. Amino acids and vitamins are there for you in paper 2, more particularly CH 202 organic chemistry. So vitamins. Okay, maybe what I will do, I will continue this with vitamin chapter. So after the completion of this chapter, I will take one more chapter called amino acids, and later on one more if possible. Okay, that is about syllabus for this semester. Uh, uh, okay. So with this, I will go ahead. Maybe now I will switch over to presentation mode. You just tell me whether you are able to see the cursor or not, pointer or not. So this is presentation mode. I will take a pointer now. Pointer. Okay. So now pointer is visible or not? So the pointer is near to the vitamins.
Not visible, sir.
Not visible. Okay, I will go to the next slide. Okay, next slide is visible or not?
Not visible, sir.
No, sir.
Vitamins. Vitamins. Visible. Slide. I moved a slide.
It's not visible, sir.
No, I am talking about the. I just moved one more slide. What you are able to see there?
It's on
First
the
slide
intro
only,
page
sir.
only.
First page or second page?
First
First.
page.
Introduction, sir.
Introduction. No, actually, so now I moved. Actually, I was in this. What is the vitamins classification? Vitamin A, B. So all those things is there as per my. Uh, Introduction here, there is no introduction, no? Only one introduction is a third page.
First slide, sir.
First slide, it's only the
Yes,
name of the paper, no? Paper and the
yes, sir.
vitamins is there. So now what you
That's it.
can, okay. So I'm moving to the second slide. Is it moved?
No, sir.
Okay, fine. Maybe I think so. I will start with this. Now again came back to the first slide. Now tell me the cursor or pointer, laser pointer is visible or not?
No, sir.
Okay, if the laser pointer is not visible means I will take a pencil. You just look into that. Now you see. Okay, now I took a pencil and now I am marking near to the vitamins. Are you able to see that?
No, sir.
No, sir.
No marking below the vitamins?
No, sir.
No. So that means I won't go for a presentation mode. I will come back. Okay, now you tell me it's not in presentation mode. Now could you see the movement of cursor pointer?
Yes, sir. Visible, sir.
Okay, right, fine. Maybe like this only I will explain it. Uh, So the paper name is CH, the paper code CH 202. Okay, and the name of the title of the paper is Organic Chemistry. Under that, the unit 4, okay, contains a chapter name called vitamins. Okay, I am taking that chapter. So with this, I will go to the next slide. So next slide is what and all it contains, the content of this particular chapter. So in this chapter, we will discuss about vitamins. What are the vitamins? Is somebody is waiting? Yes, I will. Okay. So in the we are discussing about these vitamins and how they are classified, and we are discussing about the vitamins called vitamin A. This vitamin A further divided into vitamin A1 and vitamin A2. And we will also discuss about the vitamin B2 is called vitamin B1. No, vitamin B1 is called thiamine. Vitamin B2 is also called as riboflavin. Vitamin B3 is also called as pantothenic acid. Vitamin B6, it's also called as pyridoxine. Vitamin H is nothing but a biotin. Vitamin M is nothing but a folic acid. Vitamin C is nothing but a ascorbic acid. Okay, and we will also discuss about vitamin E. It's called tocopherol, and vitamin K is also called as phylloquinone. Phylloquinone or hydroquinone is nothing but a what is called as vitamin K. So then, so with this, I will go to the with this I will go to the next slide. So it's about introduction. Okay, right. So introduction means what are vitamins? Vitamins are organic compounds that are required uh, for normal growth and proper nourishment of the body. So that means vitamins are organic compounds that are required for normal growth and proper nourishment of the body. In addition to the fats, carbohydrates, proteins, minerals, salts, and water. Okay, these are also organic compounds. Okay, then if you want to write about the definition for vitamins, means you can say that vitamins are the organic compounds that are required in a small quantity for normal growth and proper nourishment of the body. So without the supply of vitamins, the individual will develop nutritional deficiency diseases. About 20 different vitamins are known. Those are denoted by alphabets A, B, C, D, E, K, etc. H, okay, M also. Okay, I think somebody is there. Let us make in. Right. Uh, okay. So, without the supply of vitamins, the individual will develop a nutritional deficiency diseases. About 20 different vitamins are known. Those are denoted by alphabets, more particularly A, B, C, D, etc. Each of these vitamins plays a specific role. Each of these vitamins plays a specific role, and no other compound can act as a substitute. And no other compound can act as a substitute. Some of the vitamins like B3, okay, some of the vitamins like B3, okay, B3 is nothing but a pentothenic acid. And what is the H? It's called a biotin. And K is called phylloquinone. That means vitamin B3, vitamin H, and vitamin K are produced by bacteria. Okay, vitamin B3, vitamin H, vitamin K are produced by bacteria in the intestine and are produced in reasonably in and that are produced in reasonable quantity to meet the body's requirement. That means it only produce only the body's requirement quantity, not the more. But the rest of the vitamins, okay, other than B3, H, and K, remaining vitamins must be supplied in the diet. So vitamin D is supplied in fruit or may be produced in the skin by irradiation of sterols. I think you know that. So our skin synthesizes vitamin B, vitamin D when the skin is exposed to sunlight. Okay, right. Anyway, when we are discussing about vitamin D, we will explain more about this. And more particularly in cities like mega cities, okay, those who are leaving the house early in the morning before sunrise, and those who are returned to the house after the sun set, means after 6 o'clock, for them it is difficult to expose to the sunlight, okay, and that is also a deficiency of vitamin D. Okay, more particularly we can say that in children it's rickets. Okay, maybe something like bow-legged arms. Okay, right. Then maybe in adults one can end up with the osteoporosis. Okay, right. It's bone related one. So bone should be right. Okay. Anyway, let's not go into the too many details. What we need to take it out, the vitamin D is synthesizes when our skin is exposed to the sunlight. So that's what uh, we will note at this moment. So many vitamins have now been isolated and their structures established. Okay, many vitamins now have been isolated and their structure established. So with this, I will go to the specific example. So that is before going to the specific example, let's look into the classification. So classification is based on the solubility. If the vitamins are soluble in 1 minute, okay. Uh, if the vitamins are soluble in water, then those are called water soluble. If the vitamins are soluble in uh, other than water, it's called fat soluble ones. Okay, so water soluble ones are called vitamin B1, it's called thiamine. Vitamin B2, it's called riboflavin. Vitamin B3 is called pantothenic acid. Vitamin B6 is called pyridoxine. Vitamin C is called ascorbic acid. Vitamin H is called biotin. And these are the water soluble vitamins. And the fat soluble vitamins are vitamin A1 and A2. So vitamin A1 is called retinol. Vitamin A2 is called dihydrogenol. Vitamin E is called tocopherol. Vitamin K is called phylloquinone. Okay, so with this, I will go for a specific example called vitamin A. So, vitamin A comprises two, that is A1 and A2. A1 is called retinol, A2 is called dihydroretinol. So now let's look into the source. Okay, source of vitamin A1. Okay, so the source where and all you can find the vitamin A1, more particularly the source. The vitamin A1 we can get it from the milk. It is also possible to get it from the butter. It is also possible to get it from the eggs. Okay, and it is also possible to get it from the liver. Okay, so that means liver also contains vitamin A. Fish liver oil. Okay, I think fish liver oil is nothing but a cod liver oil. It is available in the medical shop itself. We can get it. And sweet potatoes. Okay, potatoes also contain vitamin A1. Green and yellow vegetable oils. Okay, more particularly you can find more maybe the spinach. Okay, not only the spinach, all green and yellow vegetable contains vitamin A1. Okay, then what are the deficiency effects? Suppose vitamin A deficiency effects means night blindness. Okay, during the daytime it's okay with respect to the sunlight, it's visible, more visible in presence of sunlight, and maybe in the night time visibility decreases. Okay, uh, anyway, when we are discussing about the biological importance of vitamin A, we will also discuss about that. So now the deficiency effects of vitamin A is called night blindness. So how it is causing? It is causing by drying and thickening of the mucous membrane that covers eyeball. Okay, which may lead to total blindness. Okay, this is about deficiency effects. And what is the structure of retinol or vitamin A is this one. Okay, so what you can observe that what a double bond here, this also a double bond, this also a double bond. So you can observe that all these are conjugated double bonds, and you can also observe that all these double bonds are in trans. Okay, not the cis one. Okay, during the biological importance we will talk about trans. Okay, more particularly at the 11th position. Okay, how where you are starting? You are starting with numbering from here. Okay, you can continue like this. Okay, right. uh, Next, this is the structure, and retinol also this is only. So with this I will go to the next slide. Next slide I will discuss about the synthesis of vitamin A1, nothing but a synthesis of retinol. So I will go to the next slide. So next slide, you can observe that this is name of this compound is called ionone. Okay, ionone, more particularly it's called beta ionone. Okay, that means synthesis of vitamin A1, it is possible to synthesize starting with the beta ionone. So then how we can uh, you know that this is a carbonyl carbon and this is a carbonyl carbon. Carbonyl carbon is electron deficient. Okay, and what is the thing you need to remember here? So in presence of base, okay, so there will be a formation of anion here. So that anion form a bond with this carbon. Okay, so more particularly this it's a name reaction. What is the name of this reaction? Maybe I think glycosidic epoxidation, Dargen's glycosidic ester synthesis. Okay, so this reaction is based on the Dargen's glycosidic ester formation. So how the Dargen's glycosidic ester formation? Uh, it will react with the a compound. What is the name of this compound? This is an ester. This carbon is called alpha carbon, alpha halo. Okay, alpha halo esters when it is treated with the metal. Okay, so what happens? This carbon. The obtained anion react with the electrophile. Here the electrophile is carbonyl carbon. When there is a new bond formation between this carbon and this carbon, so that time this bonded pair attack over here. Why it is attacking? Because Cl is going out. So that is how the epoxide is formed. Okay. So this is a name reaction. If you want to a detailed mechanism, you just look into the Dargen's glycosidic ester formation. So a ester is formed. So this is a epoxide, okay, and this is a ester. Okay, this method of preparation is called Dargen's glycosidic ester preparation. So from here to here, it's a three steps reaction is involved. Okay. What is the first step? First step is the ester hydrolysis. This is a ester, no? C2H5 is breakdown into a acid. Means ester is converted into acid when it is treated with a proton source. The proton source may be a sulfuric acid one or maybe a HCl one. Okay. So what will happen? The lone pair present on this oxygen picks up a proton and there will be a breakage between the oxygen and the ethyl group. Ethyl will go out as a ethanol forming a OH. That is about when what is the acid is obtained when it is treated with hydronium ion. Hydronium ion is nothing but a acid hydrolysis. In the second step, okay, after the acid formation, just by heating, so there will be a removal of carbon dioxide. How the removal of carbon dioxide? The bond between the O and H means that bond pair is coming over here. This bond pair will come over here. This bond pair will come over here. This bond pair picks up the proton. So that is how a compound, a aldehyde, more particularly alpha beta unsaturated aldehyde, okay, is obtained. This is again another key compound in the synthesis of vitamin A. Okay, you know how the glycosidic ester is converted into alpha beta unsaturated aldehyde. So now so a Grignard reaction. Grignard means this is a nucleophilic in nature. The bond a C minus, it will be available as a C minus and Mg plus Br minus. So this bond pair form a new bond with this carbon. So that means CHO is gets converted into OH. And there will be a new bond formation between the this carbon and then this carbon. So that is how this product is obtained. So from here to here, it's a reduction. So more particularly, a triple bond is reduced to double bond. Okay, the conversion of a triple bond to double bond is called uh, Lindlar catalyst. Okay, Lindlar catalyst is the one which reduces the which convert the triple bond to double bond. Nothing, and further reduction won't happen. That is the speciality of the Lindlar catalyst. So this triple bond is converted into double bond. That's all. Only that changes. Where is there is a changes, you concentrate on that. And remaining thing you just retain. Okay, that is the thing you need to do it when you are writing the scheme of synthesis of vitamin A or any other compounds. So now here from here to here, how it is changes. You know that O hydroxy group. You also aware about the living group. Okay. So living group means the one which leave the place. Nucleophile means the one which is attacks. You normally nucleophiles are electron rich one. In the living group there are different kind. A better living group, poor living group. Okay, such kinds are there. All these things comes under nuclear mechanisms. Okay, reaction mechanism. So here OH is not a better living group, but OH when it is O acyl. O acyl is the better living group. So to make this OH as a living group, this OH is treated with acetic anhydride or acetyl chloride to convert OH into a O acyl. So when OH is converted into O acyl in the first step, when it is treated with acetylating agent, uh, then what will happen when it is treated with the base? So this base picks up the proton here, and this proton is called allylic proton. Why it is called allylic proton? Because this proton is adjacent to the double bond. Okay, that is the reason when the base picks up this proton, this bond pair is coming over here, this bond pair will come over here, and this O acetyl will go out. That is how a vitamin uh, uh, A is obtained. Okay, now tell me, is there any doubt in this? The synthesis of vitamin A1. Do you find any difficulty? If you are not able to understood, I will explain. Hello.
Yes, sir.
Uh, uh, any doubt in this? The synthesis of vitamin A1?
No, sir.
No, sir.
Okay, right. So with this, I will go to the next slide. Next slide is about the synthesis of A2. A2 is nothing but a dehydro retinol. So you should be aware that the difference between the A1 and A2. So A1 means this. A2 means there will be additional double bond here. So that's all. Only the additional double bond present over here, it's said to be a vitamin A2. Okay. So I will move to the next slide. Synthesis of vitamin A2. Okay. It's also called as dehydroretinol. Okay. So this dehydroretinol means only the presence of double bond. Okay. So without this double bond is called retinol. With this double bond is called dehydroretinol, and this one is A2. Okay, you make it as this A1 as A2. Okay, then uh, okay, how it is? So what then? What is the source? Okay, if we are looking into the source, liver of saltwater fish. Okay, liver of saltwater fish is the source of dehydroretinol. Okay, I think you are aware about the difference between the saltwater fish and freshwater fish. Okay, the fishes which are present in lakes, those ones are called freshwater fish. The fish which is present in oceans, okay, is called a saltwater fish. Only the saltwater fish one contains dehydroretinol one. Okay, so now the first step, only the first step is changes, but the remaining steps are same as per the vitamin A1 synthesis. Okay, that's what you need to remember. So only the uh, step 1 is different when it compares to the vitamin A1 and A2 synthesis. Okay, so what is happening here in the first in the retina in the A1, beta ion was taken, whereas in A2 beta ion itself is taken, but it was treated with NBS followed by heating, followed by NBS. So what it means? It means that you know that this carbon and is called allylic carbon, and this carbon, this proton is called a allylic proton. So allylic proton can be uh, replaced with the bromine when it is treated with NBS. NBS is the N-bromosuccinamide. It is a reagent. It will act as a source of Br plus. Okay, that Br plus form a bond with this proton and forming a N-bromo. After formation of bromo here, when it is heated with the base, there will be a removal of dehydrohalogenation. That means one proton from here and Br minus will go out. There will be a formation of double bond. Okay, this process is called bromination followed by dehydrohalogenation. That is the formation of alkene. Alkene means double bond. The first step is this one. Okay, then afterwards next is same as previously I explained in synthesis of vitamin A1. No, that process, that the steps are same. So here this is called uh, dehydro. Okay, dehydro beta hyanon. Dehydro beta hyanon, when it is treated with alpha halo ester, undergoes Darjan's glycosidic ester formation. So here Darjan's glycosidic ester formation means the metal react with the alpha halocarbon, forming an anion. That anion react with the electron deficient carbonyl carbon, and this bonded pair. Okay, this bonded pair again attacks the chlorine because chlorine will go out. So that is how the epoxide is formed. This is an epoxide, and the obtained product is called glycosidic ester uh, product. And in the next step, it's a three-step reaction. The first step is ester hydrolyzed to acid. Second step is just by heating gives a decarboxylation. That means the bond between the O and H is coming over here, and this bond will come over here, and this bond will go over here, and this bond picks up that proton. Here one proton was there, no, that picks up proton, forming a alpha beta unsaturated aldehyde. And this alpha beta unsaturated aldehyde, when it is treated with a Grignard reagent, more particularly C minus, means anion here. Okay, magnesium adjacent to this magnesium will have a anion. Magnesium is positive. Br minus, Br is a negative. Magnesium is having a 2 plus, one this side, another this side. Okay, so this carbon is anion that it will act as a nucleophile. It is electron rich species, and it will form a bond between this carbon and this carbon, and aldehyde is gets converted into alcohol. So that is how this product is obtained. Now there is a triple bond. This triple bond is gets converted into double bond by treating with Lindlar catalyst. Lindlar catalyst is nothing but a hydrogenation in presence of palladium. So this triple bond is gets converted into double bond. Then afterwards, what will happen? OH is a poor living group. This can be converted into a better living group by treating with by acylating or by treating with the acetic anhydride or acetyl chloride or acetylating agents. So OH is converted into acetoxy group. Means acetoxy group. Acetoxy group is a better living group. So when this acetoxy is treated with a base, the base may be OH minus or maybe pyridine or maybe pyrrolidine. What will happen? The base abstract this proton. That time the bond pair is coming over here. This bond pair will come over here, and this will go out. So that is how the vitamin A2 is obtained. This is not A1, A2. You have to make it as a A2. Okay. If I'm able to convert this into a presentation mode, there I can take a pencil and I can correct it. Now it's difficult. Okay. This is about how the vitamin A2 is synthesized. So now I will move on to the biological importance of vitamin A. So this is the biological important. I think you know that the mechanism of vision. Okay, the application of biological importance of vitamin A is nothing but a vision. Okay, mechanism of vision more particularly. So our eye, okay, eye contains retina. Retina, retina of the eye contains cone cells and rod cells. So that means retina of the eye contain cone cells and rod cells. Cone cells is responsible for the vision in color and light. That means cone cells is responsible the vision in presence of sunlight and in presence of color. Color identification and light identification can be done by cone cells, whereas the rod cells is are responsible for the vision in dim light. Dim light means during the night duration. So rod cells are responsible for the vision in dim light. Then then chemically what is happening? So in rod cells, vitamin A is oxidized to aldehyde. Okay, so in rod cells, vitamin A is oxidized to aldehyde. So you know that vitamin A is contains hydroxy group. That hydroxy is converted into aldehyde. So this process occurs in rod cells. And also the trans double bond in vitamin A, the trans double bond at 11th carbon atom is polymerizes to cis. Okay, two things is happening in the rod cells. In rod cells, uh, hydroxy group is converted into aldehyde, and at the same time 11th double bond trans is converted into cis. Okay, this is what happening in rod cell. Then the protein, okay, the protein opsin. Okay, I think proteins are nothing but a peptide, isn't it? So more particularly, for this peptide or for this polymer, the monomers are amino acids. So amino acids means there are 20 amino acid. Anyway, regarding amino acid, we will discuss in one more chapter name called amino acids and proteins. Okay, there I will discuss about more on amino acids and protein. But at this time, what I will explain that opsin is a protein. That protein is made up of amino acid, and in that amino acid, we know one amino acid called lysine. Lysine is the basic amino acid. It contains a side chain called butyl amino group. Okay, amino, that amino will act as a base. So here what is happening, the side chain part of the lysine of the opsin protein form a bond with the double bond. That means it will form a bond with the aldehyde, forming an skip base. That skip base is called imine. Okay, this changes. What is the changes? The protein will form a bond with the what is called oxidized vitamin A. Okay, and this is called what is called as rhodopsin. Okay, opsin, protein opsin react with the oxidized form of retinol, and the obtained one is called rhodopsin. Okay, this rhodopsin, when it is exposed to the sunlight, so what will happen? The cis double bond, cis 11th position cis is converted into trans. Okay, so this transformations molecular, this change in molecular geometry cause a electrical signal. This change in molecular geometry cause an electrical signal to be sent to the brain, where it is perceived a visual image. Okay, and the trans isomer is not stable and is hydrolyzed to 11e. 11e means it's a 11 position trans retinol, and rhodopsin is that means it's releases. So here there is a formation of rhodopsin. That means opsin means a side chain amino acid of the opsin react with the the retinol, that is hydrolyzed to get back the retinol as well as opsin. Then what will happen? This reaction is referred to as a bleaching of visual pigment. Okay, this reaction is referred to as bleaching of visual pigment. Then 11E retinol is then converted back to 11Z. 11Z means 11 cis retinol to complete the reaction. This is about the what is called as biological importance of vitamin A. So what we can say that retina of the eye contains rod cells and cone cells. Cone cell is responsible for the vision in dim light and bright light. Rod cell is responsible for the vision in dim light. In rod cells, the retinal is oxidized to aldehyde. Hydroxy group of the retinal is converted to aldehyde. At the same time, 11th position, okay, 11th position of 11th position of double bond is converted into cis. These two changes occurs in rod cell. At this moment, protein opsin uses the side chain lysine amino acid side chain to form a bond with the oxidized form of retinol, and this formation is called rhodopsin. Rhodopsin is nothing but a activated complex, and this rhodopsin, when it absorbs a light, okay, there will be a change in the 11 cis to 11 trans. This molecular geometry causes a electrical signal. This electrical signal sent to the brain, where it is processed into visual image. This is about what is called the biologic mechanism of vision. Okay. Uh, any doubt up to this? Hello?
No, sir.
No, sir.
No. Okay. Maybe. Uh, right. Maybe what I will do, I can today I can stop at this stage. Otherwise, I can take one more vitamin. Okay, that means I can take vitamin B1. Okay, maybe I will do it in a next class itself. Okay, so maybe next class I will discuss about vitamin B1. Okay, it's also called as thiamine. That's all. No, if you have any doubt, you can ask me. Otherwise, means I will uh, stop at this stage. Any doubts?
No, sir.
No, sir.
No, sir.
Okay, okay, fine. Okay, maybe. Uh, okay, maybe I will stop at this stage. Tomorrow I will continue. Maybe tomorrow I will take vitamin B1 and vitamin B2. Okay, two things I will continue. I will two vitamin synthesis and their biological importance I will discuss tomorrow. Since it is a first day, I will stop at this stage. Maybe I will stop sharing. Do you find any difficulty in understanding up to this? I think could you see the screen and also you can see the cursor or pointer movement, no?
Yes, sir.
Yes, sir.
Okay, I will stop sharing now.